The story of neuroblastoma always begins with one child, one family searching for answers, one voice asking to be heard. Today, that voice is part of a growing global community. International Neuroblastoma Awareness Day was established to bring families, clinicians, and researchers together from around the world, creating a place where no family feels alone and where every question matters. Over the past year, monthly webinars have helped families and professionals learn side by side, finding clarity, connecting with experts, and breaking through the isolation of rare disease. On this second annual Neuroblastoma Day, we're building a community where information is accessible, support is shared, and collaboration drives progress. Together, we can.
Good morning, good afternoon, and good evening. We have with us people from literally all over the world. Welcome to the second International Neuroblastoma Awareness Day webinar. Today, we gather as global community, families, clinicians, researchers, nurses, advocates, and friends connected by one shared truth. Behind every statistic is a child, a family, a story that changed everything. The story of neuroblastoma always begins with one child, one diagnosis that turns a family's world upside down. But that doesn't end here, because none of us walk this journey alone. Over the last year, we've seen a growing movement, one that crosses borders, time zones, and disciplines, driven by the belief that together we can change the future of this disease. International Neuroblastoma Day is more than just a date on the calendar. It's a moment that unites people from every corner of the world. It brings together parents searching for answers, researchers pushing the boundaries of science, clinicians fighting for better outcomes, and advocates working tirelessly to ensure no family faces this journey without knowledge, hope, and community. Today's webinar reflects that spirit. We gather not only to share progress, but to build connection. Not only to discuss challenges, but to spark collaboration. Not only to remember why this fight began, but to envision where it can lead. On the second International Neuroblastoma Awareness Day, our mission is clear: to amplify knowledge, accelerate innovation, inspire hope, and strengthen the global partnerships that moves us closer to a world where every child has a real chance of a healthy future. Together we honor the stories. Together we push the science. Together we stand with every child and every family. And together we can. It is now my honor to introduce the moderator of this inspiring event, Dr. Daniel Morgenstern, the director of the New Agent Innovating Therapy Program at the Hospital of Sick Children in Toronto, and a globally recognized leader in early phase clinical trials and neuroblastoma research. Dr. Morgenstern brings a unique combination of scientific expertise, clinical insight, and deep compassion for families. His work bridges hope and innovation, helping guide new therapies from concept to clinic. We are grateful to have him here tonight with us for the discussion, to lead the discussion and help shape the meaningful global conversation. Dr. Morgenstern, thank you for moderating this event and for your unwavering dedication to improve the lives of children with neuroblastoma.
Thank you. Thank you so much, Annette, and for that very generous introduction. I hope I can live up to the expectations that have been set there. And I just want to say thank you, of course, to everyone who's joining this meeting, either live or who will hear this in a future recording. It's really a fantastic effort that has been put together to bring everyone together from across the world. And the list of speakers that we have together is really extremely impressive. We have some of the people who are responsible for leading the global consortia that drive clinical trials, as well as some global leading scientists who are pushing the frontiers of the research that helps to develop new treatments for neuroblastoma. So thank you wherever you are for joining us. In a minute, I'll give you a moment or two about the program, but I first of all just wanted to highlight on the website that you may have seen. Hopefully we can bring that picture up. There it is. So on the website, the Neuroblastoma Awareness Day website, you'll see that there is this opportunity to add a pledge, and we have a global pledge wall in development there. You can already with pins from across the globe, really. And this is a chance for you, parents, clinicians, researchers, pharma, if they're online, nurses, whoever may be on the line, to join a global community in acknowledging the importance of the work that everyone does for neuroblastoma, be that raising awareness, support for families, support for research, whatever your particular interest is. So you'll see the QR code there, and you'll see the pins that we have on there at the moment. And I just encourage you to take a moment during the meeting to add your own pledge there, whoever you are and whatever your involvement in neuroblastoma. So if everyone's had a chance to take that QR code, we'll put it up again later as well. And if you can't find the QR code, then you can just go to the website to search. We're going to get into the first of today's sessions. So the way this is going to work is that the first session is about clinical trials in their biggest context. And we have, as I mentioned at the beginning, some great speakers from the leadership of the big international trials group that run the clinical trials that really help to push our treatment for high-risk neuroblastoma especially forward. And then after they give their talks, there'll be an opportunity for questions from you, the audience. So hopefully you'll see on your Zoom screen a little square down the bottom called Q&A. And if you click on that, there's the opportunity to put a question in there that we can then address in the Q&A at the end. So anytime you have a question that comes up, please feel free to put it in there, and we'll then address those questions all together at the end with our speakers. And then after this session, we'll move into another session looking at some of the underlying science that's advancing treatment or monitoring of disease for patients with neuroblastoma. And I'll introduce that in a moment. So without further ado, let's crack on and introduce our first speaker, who I'm very pleased to say is Dr. Bagatell, who's a professor of pediatrics and a neuroblastoma oncologist at Children's Hospital Philadelphia. And most importantly, in the context of this meeting, is the chair of the neuroblastoma specific committee under the Children's Oncology Group, which, as she'll talk about, is one of the big global trial consortia. So Ro, many thanks for joining us. The floor is yours. Thanks.
Thanks, Daniel. I was saying to Daniel just before we started that he has a future career as an MC. He's extraordinary at that. And I've always, my whole life, wanted to say crack on, or have. Now you have. Okay. Can everyone see my slides?
We can.
Okay, great. So I followed the template here, and I hope I hit the points that people are really interested in. I'll try to keep this relatively brief today because I just wanted to address the questions that seem to be of most interest to the organizers, but happy to answer any questions from the group after that. So I was asked to provide a little bit of a bio. So yes, I'm at the Children's Hospital of Philadelphia. I'm a pediatric oncologist. I lead the solid tumor group here at CHOP, and my area of interest, of course, is neuroblastoma. I have the pleasure of also leading the COG Neuroblastoma Committee, which is one of the most challenging and yet rewarding things I've ever done. I've done a lot of clinical research over the years and have led a number of clinical trials. My favorite part of my academic activities is really engaging with people who think differently, whether they're surgeons or radiation oncologists or laboratory scientists. I find that multidisciplinary engagement and challenge really quite intriguing. But most important of everything I do is take care of patients and work with families. And I see a lot of kids with neuroblastoma. And I know that some of the people who are listening to this are parents of some of my patients. They told me they were watching, so hi. Just a little bit, I think most of you know what the Children's Oncology Group does. It's over 200 institutions, primarily in the US and Canada, but a few in Australia, New Zealand, and one in Saudi Arabia, that all come together to try to pool resources and ask and answer questions about pediatric cancer. COG itself is not only focused on neuroblastoma. Neuroblastoma is one of the disease committees within COG. COG is the umbrella organization, and then we in the neuroblastoma committee are laser focused on this particular disease. But it turns out that more than 80% of children diagnosed with cancer in North America are treated at COG member institutions. And although those institutions open a variety of trials for children with different kinds of cancer, we are very fortunate that these institutions elect to open high-risk trials for children with neuroblastoma. So our ability to enroll patients on trials and learn from every child is really tremendous. There are about 12,000 patients enrolled on COG trials per year, and the accrual on our neuroblastoma studies, as I said, tends to be quite brisk. I was asked to talk a little bit about how COG is set up in general and how the neuroblastoma research infrastructure is set up in particular. So at the very heart of everything we do is a desire to learn more about neuroblastoma, particularly the biology of neuroblastoma, so that we can come up with better trials and new therapies. So at the heart of our research are these trials called biology trials. They're not treatment trials, but the predecessor was set up a number of years ago, about 25 years ago, by Sue Cohn and others. It was the ANBL-OOB1 trial, which was a neuroblastoma-specific biology study whereby patients would register and certain basic information would be collected about those patients. They would have tumor testing and then tumor banking, and then their progress over time would be followed so that we would have information about their risk classification, a little bit about treatment, and then their progress throughout the course of their therapy and follow-up. What's really important about that is that the data from these studies initially just built the database, but then over time, we were able to learn from those brave early patients and families who consented for this study to really follow those patients, look at the biology of their tumor, and come up with modified guidance regarding risk stratification. So what we know about neuroblastoma risk grouping today came from the early cohort of patients in the 2000s, and we're constantly leapfrogging and learning from patients as we go. This is particularly important because we want to understand our patients' characteristics as best we can. And as many of you know, we use a lot of prognostic factors in neuroblastoma to inform our therapy selection. And all of that comes from the biology studies. The ANBL-OOB1 specimen bank has been an incredible resource for scientists, laboratory-based scientists around the globe. And what makes it so special is not just that there are a lot of tumors in there, which there are, but that these specimens are associated with really important clinical information so that the laboratory scientists are not just testing the samples, but they are informed by the clinical course of the patient. And that has proven invaluable over the years. The ANBL-OOB1 study was so successful that in many ways, other disease groups patterned what they do on what neuroblastoma was doing. And to a real extent, the now disease-wide, COG-wide study APEC-14B1 is a new and improved biobanking study, but largely patterned on the neuroblastoma model. The other really important piece of infrastructure to know about COG is that there's a common database. It's called RAVE. It was not developed specifically for neuroblastoma or even for COG, for that matter. It's used across the National Cancer Institute clinical trials groups, and there are pros and cons for that, about that for sure. It does, however, facilitate uniformity of data across multiple trials, so we can compare the data collected from one trial to another within the U.S. National Cancer Institute system. It is separate, however, from the databases of other international cooperative groups, the legacy German group, SIOPEN, the UK groups, the Japanese groups. So we would love to have easier communication within the databases, but that's not really how those databases were set up. We're all very fortunate, I think, and I know the leaders of the other groups would be the first to acknowledge the important work of the INRG. where data from the COG database and SIOPEN and others can be contributed to a unified database so that we can all learn from each other. But the way the databases were originally set up is in fact separate. But I do think there's a big effort towards harmonization in the neuroblastoma community in particular. I don't know if this is evident to parents, but it is an incredibly collaborative community. I think that the INRG work and other kinds of scientific collaborations have really set the tone for collaboration. And there's been incredible work, I would say, over the last 10 to 20 years to harmonize what we do, make it complementary, and really communicate well about what we're doing. A perfect example of that was the original INRG coming together to all use the same approach to risk stratification. I think the international approach to pathology classification is another example. And another one is the International Neuroblastoma Response Criteria that we share across cooperative groups. So there are a lot of ways that each of the groups operates independently, but there's collaboration even despite those separate setups. The next thing I was asked to address was the areas of focus for the coming year. I can't believe 2025 is almost over and 2026 is upon us, but so it is. And the areas that I understand are of particular interest to this group are immunotherapy, MIBG, the future of high-dose chemo with stem cell rescue, and then other kinds of strategies to address the problem of relapse. So I'll start with immunotherapy. As many of you know, there was an incredible shift in paradigms a number of years ago, 20-something years ago, towards using the body's own immune system to address minimal residual disease in neuroblastoma. And what we found out through the ANBL 1221 study was that you could really enhance the effectiveness of immunotherapy by combining it with low-dose chemotherapy. And there were some quite important studies conducted at St. Jude and a pilot study in the Children's Oncology Group, ANBL 17P1, studying the early incorporation of chemoimmunotherapy for high-risk patients to try to prevent rather than treat relapse. That led to the ANBL 2131 trial, which is currently open, and accrual to that study is brisk. People are really interested in that study and enrolling on it, and we anticipate completion of that trial in 2027. I'll talk next about what comes right below that. So the next trial will be taking this to its logical next step, which is asking the question and answering with data, the question as to whether in the era of immunotherapy, we may be able to avoid the toxicity of high-dose chemo with stem cell rescue. So the idea would be to study in patients who have responded to treatment in a randomized way, where half the patients get one therapy and half get the other, is to use 6 cycles of chemoimmunotherapy versus 2 back-to-back rounds of high-dose chemo with stem cell rescue, and really answer the question once and for all of whether we still need high-dose chemotherapy with stem cell rescue in neuroblastoma. The initial concept for this randomized trial was submitted, and our funding agency, which is the National Cancer Institute, did not approve it because of some concerns about the statistical design. However, we are undaunted, and we've actually gone back and really thought about ways to address this problem and have come up with what we think is a really rigorous and important statistical design. And the new concept will be submitted in early 2026. My opinion is that this study needed to open yesterday because this is the question that everyone really wants to know. And I know that there are parents listening to this talk right now who are making this decision right now. What do I do with my child who's responding to treatment? Do we still need high-dose chemo with stem cell rescue? Is chemoimmunotherapy a really viable and well-supported option? And the answer is that we need to do the trial to find out. So I have incredible enthusiasm about this study, and I hope that you do too. To round out the information on this slide, MIBG is another really important strategy for the treatment of high-risk neuroblastoma. And based on the data from studies in patients with relapse and refractory disease, we in the COG asked the question of whether early administration of MIBG could, again, prevent rather than treat relapse. And so we ran the ANBL 1531 trial, which was completed in 2023. And next year at this time, we'll know the answer as to whether early administration of MIBG improves outcome. Nobody knows the answer at this time, and so we're just eagerly awaiting those results. We'll present those internationally in the spring of 2027.
Thanks, Ro. We're a little bit at time, so
okay.
We might talk about the relapse section in the Q&A, if that's okay.
Sounds great. Yeah. Sorry, I thought I was going to be faster.
That's all right.
I'll round this out just by saying we really are interested in new designs, more efficient designs. ANBL 1531 was originally designed as an adaptive study. Unfortunately, our funding agency did not approve that, but we did use a semi-adaptive design, and what we found was that one arm could be dropped early. So this semi-adaptive design really worked and was more efficient for our patients. I was asked about the use of historical controls. We have proposed the use of historical controls, but the relatively conservative reviewers and regulators that have to approve our concept did not approve the use of historical controls. So change is coming, but it's quite slow. I think the answer to moving the field forward faster is through collaboration. And I'm really optimistic about our ability to work together with our colleagues around the world to run more efficient trials and trials that answer different questions. So in looking at the road ahead, I will say that collaboration is pretty much the main word I'd want to leave you with. Collaboration with our colleagues, of course, who are interested in neuroblastoma, collaborations with patients and families to identify the most pressing questions, and then collaboration with regulators on all levels to try to help us all understand what new statistical designs are appropriate, what new controls are appropriate in order to move forward faster. Industry has been incredibly collaborative with us in the last few years, which was not always the case. And this is especially important right now due to funding cuts in North America. COG is having to do more with less. And so collaborating with our industry partners is absolutely essential, even more than it used to be. I think that some of the work we do as clinical trials organizations is incredibly tedious. And my hope is that we'll be able to use AI to move faster and get more done. There'll be some talks later on about new technologies, imaging techniques, ctDNA. In the COG, we're all over that and have been incorporating those new technologies into clinical trials. And finally, I do think that we have really prioritized in the COG Neuroblastoma Committee some studies of social determinants of health. We've clearly shown that it's feasible to collect data about social determinants of health, race, ethnicity, socioeconomic factors, and understand where the disparities come from. But now it's time to intervene and try to address some of those things. So we're very committed to that. I'll stop there.
Thanks so much, Rose. So thanks to everyone for putting your questions in the Q&A. We are seeing those and we're going to address those all at the end. So we're going to move now to Dr. Julie Park, who I think unfortunately is joining us via video in a recording because she has a conflict at this moment. And so she's a pediatric oncologist at St. Jude Research Hospital in Memphis. And most importantly, in the context of this meeting, is the scientific chair of NANT, which is the New Approaches to Neuroblastoma Therapy, the early phase trial consortium for neuroblastoma. So hopefully we have Dr. Park's video that we can play. And I'll say roll VT.
I'm chair of the Department of Oncology at St. Jude Children's Research Hospital, and I'm scientific director of the New Approaches to Neuroblastoma Therapy Consortium. I'd like to thank the organizers for having me present to you today. I apologize that I'm not able to be there to present live, but appreciate the opportunity to send this recording along to you. NANT was founded in 2000 as an idea from Bob Seeger at Children's Hospital of Los Angeles and Kate Mathey at the University of California in San Francisco. And their thought was that by bringing together laboratory investigators with clinical research investigators, we could more rapidly evolve novel therapies for children with high-risk neuroblastoma. Since then, NANT has grown to 14 institutions within the United States, 1 in Canada, and 4 institutions in Europe and UK. Kate Mathey and Judy Villablanca. Judy was the original medical director of the Operations Center at Children's Hospital of Los Angeles, remain involved in the operations of NANT. And Araz Marichelian has taken over from Judy as our medical director for the CHLA Operations Center for NANT. NANT was really founded on the idea or the importance of translating laboratory discoveries into clinical trials. It's the first consortium solely dedicated to innovative clinical trials for relapsed and refractory neuroblastoma. It's a unique collaboration of laboratory and clinical investigators that meet on a regular basis to drive our translation of discovery into clinical trials. With this collaboration, we've enrolled 850 patients on therapeutic clinical trials. 650 patients have enrolled onto our biobanking trial, allowing for patient material to be available to laboratory researchers for further discovery. We enroll about 40 patients a year onto our therapeutic trials, and currently we have 2 active trials with the hope of opening 2 additional trials in the next year. Our areas of focus are really currently divided between looking at how to enhance immune therapy for high-risk neuroblastoma or how to enhance MIBG therapy for neuroblastoma. Anti-GD2 antibody therapy has become a mainstay of treatment, both in the upfront setting as well as in the setting for treatment of children whose neuroblastoma has been resistant or relapsed after upfront treatment. But we know that anti-GD2 antibody is not effective for all children with high-risk neuroblastoma. In fact, only about half of children with relapsed neuroblastoma will respond to the combination of chemotherapy and anti-GD2 antibody. So through our laboratory investigations, we're trying to understand ways to enhance the activity of this antibody. And one way is to improve the numbers and activity of NK cells, which are important immune cells that are necessary for the function of anti-GD2 antibody. In an earlier study run by Araz Marichelian, we established that we could collect NK cells from patients, send them to Columbus, Ohio to be expanded, and then back to treating sites to give in combination with anti-GD2 antibody. But we also found that the cells from patients were very difficult to expand and did not have as much activity as we would have expected. We think this is because patients with neuroblastoma, especially those with relapsed neuroblastoma, have an immune system that has gone through a lot of therapy and may not be as effective as an immune system from someone who has not undergone chemotherapy. So for that reason, we are now collaborating with Dean Lee and Carrie Strebe at Nationwide in Columbus, Ohio, to use donor-derived NK cells that can be expanded and then delivered in combination with chemotherapy and anti-GD2 antibody. That trial is now ongoing in the United States across the NANT consortium. Similarly, Sarah Federico at St. Jude is running a clinical trial at NANT sites under the sponsorship of St. Jude that is looking at an IL-15 superagonist in combination with chemotherapy and anti-GD2 antibody. IL-15 is a cytokine that is known to be important to increase numbers of NK cells and activity of NK cells. And this will be a randomized trial to understand whether the addition of IL-15 superagonists will improve response to chemotherapy and anti-GD2 antibody. And finally, through investigations of performed at Princess Maxima and in the United Kingdom. We will be looking at new therapeutic targets against immune checkpoint signaling that prevents the immune system from actively engaging with the anti-GD2 antibody. And we hope by inhibiting those immune checkpoint signaling pathways, we can improve the efficacy of chemo and anti-GD2 antibody. We'll be first running this smaller clinical trial to primarily evaluate the safety of adding these immune checkpoint inhibitors in combination with chemo and anti-GD2 antibody. And hopefully, if we see a signal of response and we see safety, we can bring that to a larger clinical trial within COG or the SIOPEN consortium. Investigators at Children's Hospital of Philadelphia are also looking at novel cellular therapies, looking at genetically modified T cells from patients to recognize neuroblastoma cells and bring immune cells directly to the neuroblastoma cells for anti-tumor activity. There are 2 trials that are ongoing there. These are first-in-human early phase trials that are being done only there to evaluate early safety. If the safety trial is successful and we show that we can deliver these without unexpected complications, we hope to expand those trials to larger trials across the NANT consortium. And then finally, for MIBG, we have shown through multiple trials in the NANT consortium that MIBG can be effective in about 30% of patients with relapse or refractory neuroblastoma. But obviously, that's not enough, and many of the responses are not durable. So we're again looking at various combinations that we hope will enhance the activity of MIBG, and we hope to be opening a randomized phase 2 clinical trial to look at whether the addition of anti-GD2 antibody or the addition of vorinostat or the addition of both together will improve response rates following I-131 MIBG. And then finally, we will be opening this year a master biomarker trial called MIGHTY that will look at molecular and immunologic targets across the spectrum of our clinical trials done serially so that we can really understand what is happening to the patient's immune system and what is happening to the tumor in response to therapy. We hope that this will help us to be able to better identify biomarkers of resistance, identify how the tumor is evolving, identify better ways to assess response, and hopefully in the long run, identify better precise diagnostic tools to help us deliver the best therapy for each individual patient. Collaboration is really the mainstay of NANT. We're an international collaboration. Its real foundation is academic collaboration. As I said, this was built on the idea of laboratory investigators working with clinical investigators. This has been funded since 2000, but through the National Cancer Institute, through a program project grant, and through additional funding from foundations, both in Europe, Canada, and the United States. With that, we also need to develop partnerships with biotech and pharma to have access to therapeutics that we can deliver in our clinical trials. We work closely with companies to share preclinical model testing of agents and work with them to design clinical trials specific for neuroblastoma so that we can quickly translate our findings in the preclinical setting to early phase clinical trials. A very important collaboration is our collaboration with our patient advocacy committee. They have been an important component of the NANT consortium since its inception. They attend scientific meetings, they assist with trial development, they develop educational tools for patients and families, and they have been very strong advocates for funding for neuroblastoma and childhood cancer. And we thank all of them, in particular our chair, Susan Gusto. In the future, we hope to continue to feed into the global framework through our international collaboration. We have international meetings that include representation from COG and SIOPEN to improve our ability to translate our findings into upfront treatment for neuroblastoma, to align our biomarker analyses, and to promote sharing of both clinical and biomarker data. We hope to also align our interpretation of the International Neuroblastoma Response Criteria and to hopefully find new biomarkers that we can use to assess response. The road ahead really continues on our path of translating findings from laboratory research into new clinical trials. We have identified various mechanisms of resistance that we hope to better understand, including the regulation of GD2 expression, how molecularly tumor cells evolve during treatment, so we can understand the best combinations to prevent that resistance from occurring, and also to better understand how the immune system and tumor cells interact so that we can better use our immunotherapy and combine that with our molecular therapy to improve the outcome for neuroblastoma. With that, I'd like to thank you for your attention and thank you for your partnership and commitment to changing the lives of children with neuroblastoma.
So many thanks to Julie Park. We're going to cross the virtual Atlantic now and head to Europe. And I'm very pleased to introduce Lucas Moreno, who is a pediatric oncologist at the University Hospital Vall in Barcelona, and in this context, the president of SIOPEN, the European Collaborative Group for Neuroblastoma. Lucas, over to you. Thanks.
So hi, everyone. I hope I'll make a start and be brief, hopefully. I'm really pleased to be here, and thanks to ANAT and for the organization to have this wonderful discussion. So as I said, I'm Lucas Moreno. I'm a pediatric oncologist, currently working at Vall Hospital. I trained in Spain, then in London, and then went back to Spain to focus on the development of early phase clinical trials, but particularly in neuroblastoma and the development of precision medicine as well. It is a pleasure to be here, and I'll be giving first an overview of what we do in SIOPEN, just a touch of the clinical trials that we are conducting at the moment to share some reflections at the end. SIOPEN is a big organization. We have got more than 600 members from 27 active countries, and although we are very focused in Europe, or the organization started in Europe, we have grown recently, not only with members from countries outside Europe, but also we're already recruiting to the SIOPEN trials outside Europe, definitely in Israel for a long-standing collaboration, in Australia, New Zealand, and then with an expansion in more countries. It's a great organization to be working with, but it does bring many challenges of many different systems and regulations to run our trials and studies. For the past couple of years, we changed the structure of the organization, which is now more focused on the roles. We did a strategic review to mainly focus on clinical trial delivery and data and organization of the data projects. I'm very fortunate to work with two vice presidents, Cormac Owens and Juliet Gray, with a manager, Carla, and with the past president, Maya Beck, because all of us kind of take the lead and share the duties of SIOPEN, and we also work very closely with advocates, particularly with Nicole Scobie, Leona Knox, and Nick Bird, that started our patient advocacy committee. I'll make a brief review of the trials that we have. The frontline trial for high-risk neuroblastoma is called high-risk neuroblastoma 2. It has included now more than 700 patients from the end of 2019. It's open across countries in the European Union, Israel, and Australia, and we are still working to open more. It's the treatment is currently testing the induction, is the rapid Kojic, the induction that has been used traditionally in SIOPEN. It has an extended induction phase with chemoimmunotherapy for those patients that don't respond so well. It has got consolidation with high-dose chemotherapy, where we are testing 1 versus 2 stem cell transplants, and also asking a question on radiotherapy. We've started a pilot trial to bring chemoimmunotherapy up front, and that pilot trial is already up and running. We're close to identifying the safe dose of rapid Kojic chemotherapy with dinutuximab beta, and hopefully this will help to bring it forward into the large trial that is high risk 2. So what's going to happen next? Still within the high risk 2 trial, we will keep it open for a few more years, and we are aiming to introduce lorlatinib in a small randomized trial. The contract is already signed, and we have had a lot of help from many friends in this collaboration to make this happen. And so we're going to amend the trial and hopefully start early next year to recruit. And we are still having discussions to see if it's possible to incorporate DFMO at the end of treatment for trial patients, and also to bring a randomization following on the results of this pilot study that I mentioned, to bring a chemoimmunotherapy randomization up front. We have already started planning for the next trial, which will be the high-risk 3 trial, wanting to develop further a new international induction. Part of this collaborative work, we are trying to align treatment and efforts with internationally. So we're working very closely with our colleagues in COG. So we would hope that new international induction regimen has been defined by the time we start high risk 3. We want to evaluate new consolidation ideas, mainly to not give the same consolidation to everyone. There will be some patients that perhaps can have a less intensive consolidation, and then there would be a role for reducing or even removing high-dose chemotherapy with the stem cell transplant, whereas others may need intensification of therapy, and we're having the discussions to see what are the different ways to make this intensification. And we want to further improve the anti-GD2 therapy that is given at the end of treatment and in the post-consolidation phase, because there is still space to make this better and easier to give. In the relapse setting, we are working with the Beacon series of trials. We did the Beacon 1 trial that tested chemotherapy in combination with dinutuximab beta and bevacizumab, that had a novel clinical trial design that helped us to test different agents in a relatively rapid manner. And the addition of these drugs is already being incorporated in some of the clinical practice guidelines, although its introduction into routine care is challenging. We have also learned a lot from this Beacon trial on the molecular profile of relapsed and refractory neuroblastoma. That's the Beacon Bio project, led by Gudrun Schleiermacher, that will be speaking later, and other colleagues, where we are conducting all the molecular analysis of the samples of patients that were recruited to the trial, and also doing liquid biopsy studies, studying the tumor heterogeneity and the clonal evolution of the tumors. We're looking, SIOPEN is doing a trial of liquid biopsy in relapsed neuroblastoma. It will evaluate the role of liquid biopsy. And we have already started the Beacon 2 trial. We're working very actively. It's now recruiting in the UK and opening in the rest of countries soon. And we have done a lot of regulatory work to make sure that the drugs that are tested on the Beacon 2 trial are valid for regulatory approvals, so that we speed up access to the drugs once the drugs are tested on the trial. And because it's an adaptive design, we are exploring new arms so that we can continue to bring innovation in relapse. There are many other areas of interest where we have different trials and by different members of the group. But I think the bottom line is that we are fortunate in neuroblastoma that there's this great activity happening and lots of options. That the challenge is to bring them, to identify, to test them rapidly, and to test them in frontline as rapidly as possible. And I think to wrap up, this is a summary of a publication that we did together with our colleagues in the Children's Oncology Group and patient advocates as well, where we analyzed all the challenges that we thought with survival. The current frontline treatment is still not working for a significant proportion of patients, and we have difficulties to run the academic trials. And we really want to change this. So in our analysis, we proposed action points to deliver. So we are working on this common agenda and common roadmap. There is a lot of work going on in this front, a lot of initiatives to share data, to work together, to harmonize the work that is being done. There's still tons of work and tons of challenges to overcome, but we are hopeful that in this setting there are more new drugs, new clinical trials, interesting and important results coming up every year, and we hope this will improve the results for all the patients, and it will become available across the globe. We, as a summary, I think our bigger highlight recently is that we are working on these regulatory aspects to facilitate future access to the medicines once we finish the trials. We continue working on these challenges to open the clinical trials rapidly and to facilitate that the new drugs become available for routine care after the trials, and highlighting that international and multi-stakeholder collaboration is increasing, and it will facilitate a better future working once we're working together with across the globe. So thank you very much to a lot of people, and look forward to some of the questions. Thank you, Daniel. And
thanks so much. So thank you, Lucas, and thanks to all of our speakers. If we can put Ro on screen as well. So we're going to take some questions now. If you have additional questions, please put them in the Q&A, and if we don't have time to answer them all, we'll do our best to give you some answers in writing if we can't tackle them. So there's two kind of major sets of questions that have come up from the audience. So one of them is about maintenance therapy and what is happening and what might happen in the future about studies in the maintenance stage after immunotherapy. Are there plans for future studies with DFMO or with vaccine approach? So I don't know who wants to take that one first. Ro, I'm going to put you on the spot.
Okay. Yes. So we had actually proposed in the Children's Oncology Group to include DFMO for all patients on ANBL 2131 as a continuation phase, because we didn't want to lose the ability to answer our primary question if there was inconsistent uptake in terms of seeking out DFMO. It would just introduce another variable in that equation. Unfortunately, our amendment to add eflornithine or DFMO for all patients was rejected. So what we're doing now is collecting the data about who receives DFMO and who doesn't, in the hopes to at least learn something. Together with our colleagues in SIOPEN and our colleagues at Sloan Kettering, we had been working with the company that makes the GD2, GD3 vaccine, and had come up with, as you well know, Daniel, a beautifully, very well written, well thought out concept for a study. But unfortunately, the numbers just became prohibitive. So as much, and the company just wasn't able to undertake a study of that scale. All of us, I'm speaking for you, Daniel, more than anyone, but we really wanted to run that study on an international basis and get the answer, but unfortunately, it just was not to be. Lucas, do you have stuff to add?
Lucas, the approach in Europe on DFMO is to include it in the study?
Yeah, so at the moment, it's not approved. It doesn't have an approval in Europe, and that limits significantly the access to the medicine. There is an expanded access available in some of the countries, and SIOPEN has issued a recommendation for the time being. Our thought is similarly to what Ro mentioned, is let's include it in the high-risk 2 trial for everyone, so that at least there is homogeneous access and the results of the trials are maintained. I think still, in the best case scenario, unfortunately, there are patients still relapsing. So there's still a need for better therapies. statistically, it's a challenging area to conduct the trials if you want to test to show clear superiority. But I'm sure that there may be ways that are developing with biomarkers, with novel statistics, that there are ways to make such trials possible. And I think we need to continue working, because we definitely need to find better solutions. So the patients that, you know, with or without DFMO, the cure rates still need to improve, and the toxicities to decrease.
Yeah. So that leads nicely into the other area of questions, which was about relapse, and actually a very pertinent question about, do we actually understand what the mechanism of relapse is? What actually happens at those sites when neuroblastoma comes back? Is that new neuroblastoma, or is that neuroblastoma that was always there? What do we actually understand about why relapse occurs? Again, who wants to take that?
I can start.
Lucas, you can take that one first.
Occasion.
A share out.
I think that the short answer is that no, we don't have the full understanding of why some patients do relapse and some others don't, or do it earlier or later. There is a lot more knowledge now with the new sequencing technologies, the single cell technologies, the sequential sequencing of liquid biopsy samples. So we do know that there are different scenarios, and sometimes there are some clones of cells that remain from the beginning, remain dormant, and somehow they wake up and you kill the majority of the tumor, but that some cells are left, and then some other times there are new clones that arise. So there are different situations. We are starting to witness what is happening. And thankfully, with technology, we can look at it cell by cell or week after week, because we can monitor it in circulating DNA. We are getting to a better understanding of what happens, but we are still don't know how to change that, how to prevent or what to do. I think in the end, a very pragmatic approach is to say we need to give better frontline therapy that achieves, just kill every cell, or get rid of the neuroblastoma with the first line treatment, so that we don't have to worry about the relapse therapy.
Yeah. Because I think it'd be fair to say our assumption is that if the neuroblastoma recurs, that's because some of the original neuroblastoma cells have managed to escape, even if that was a different type of the neuroblastoma cell that happens to have remained. It's not a brand new tumor for the vast majority of patients. Yeah.
Probably not. I mean, there are some people who have a neuroblastoma predisposition.
Yeah.
Those are quite rare, but I think you're right, Daniel. And I think that in the past, we were hampered to a real extent by availability of tissue for patients at the time of relapse. But I think because of technological advancements, both in our ability to biopsy tissues in difficult to access places with our interventional radiology colleagues, that started to change. And then obviously the liquid biopsy that you'll hear more about in a little bit is a game changer. The other thing I think we need to understand is also the patients who don't relapse. Is there something different about those patients and their response to therapy? So I think we have to learn from every patient, not just a laser focus on those with bulky relapses that we could biopsy years ago, but everybody.
Yeah. And a related question is do we yet understand why some patients relapse and some don't, when, especially within high-risk neuroblastoma, they're all getting the same treatment? And why is it that some patients respond very well? Why is it some patients respond and then it recurs? Do we understand that? Lucas, you can tackle that one.
Yeah. I think our knowledge is still insufficient. We know that not all neuroblastomas are the same. There are molecular subgroups, and they are biologically different. But so that they have different characteristics: the lengthening of the telomeres, the ALK mutations, the MECAN. They're different groups, but so they behave differently. Up till now, we have been treating everyone with the same treatment because that was the best therapy that we had. I think we all would love to see that the treatment is adapted to the biology of the tumor that the patient has, and we have great hopes in that. However, we still need that will make the clinical trials even more challenging, because we will have a third of the patients or a tenth of the patients, and that's already happening with the ALK inhibitors. And we have great hope. I mean, it's still still, the biggest advances have been immunotherapy that works in all of the subgroups. So we still have to work in both fronts, in the targeted agent and in the broader therapies.
And I guess on that, sorry, Gary.
Oh, I would just add that I think when we first started to look at gene amplification and sequence aberrations, we thought, aha, we're going to understand everything. And it turns out that we understand much more than we used to, but there's so much more than DNA level aberrations that are important. I think we're starting to appreciate, at the level of RNA and protein-protein interactions, and it's way more complicated than we probably thought 15 years ago. So we just have to be a little bit humble about what we know, what we would like to know, and what we're in the process of learning.
Yeah, I mean, that's what I was just going to reflect on, was that it's probably even more complicated than just the understanding the characteristics of the cancer cells, because as you said, we know that social determinants, where you live, the family income impact on your outcome, and it's hard to believe that that is necessarily related to the tumor itself. So presumably there's a whole bunch of other mechanisms. I guess we just don't know what those are yet.
We're trying.
We are. So one final question just before we move on. One of your talks mentioned AI, and so there's a pertinent question to say, do either of you know of any projects that are actually going on at the moment looking at the use of AI in neuroblastoma, either in terms of treatment or predicting outcomes? Ro, do you happen to know of any?
Yes, I do. There are a couple that I think are really interesting, and they relate to, at this point, pathology and imaging. So those are pattern recognition kinds of endeavors, and using AI is sort of obvious in those areas. And we have projects in the COG that are starting up in both of those areas. But it's so important that we think creatively beyond just how can we use AI to improve pattern recognition? How could we use AI in trial design and the conduct of trials, the interpretation of data? I think we need some, 19 to 22-year-olds who are experts in AI to come and help us think about this, because the opportunities are endless.
Okay, so if you're a 19 to 22-year-old, put your name in the chat and we'll sign you up as someone who knows about AI. Okay, we are at the top of the hour, so I'm going to cut us off at that point. Thanks so much, Ro, Lucas, and thanks, Julie. So everyone take a big breath. That's the end of the first section. And we're going to move now into the second part of the seminar. And if you thought the first part was high pressure, high intensity, then this one is going to be even more so. So we have 5 speakers who are going to talk about their science, and they've only been allocated 5 minutes each, which is an unprecedentedly short amount of time for scientists to talk about their work. So I'm going to be extremely strict with the time. I don't know what penalty I'm going to be able to do virtually if you run over your 5-minute time, but we're going to strict timekeeping will be the order of the day. So let me stop waffling and say our first talks are going to be about CAR T-cell therapy, and we have 3 talks about CAR T-cell therapy, and then we'll move on to MFBG, which is a novel imaging modality, and liquid biopsies. So to start us off, we have Professor John Anderson, who's a professor of experimental pediatric oncology at the Great Ormond Street Hospital in London, and also UCL Institute of Child Health. And he's going to talk to us about CAR T cells for neuroblastoma. John,
Thanks.
over to you.
Thanks, Daniel. So I believe somebody else is going to operate my slides for me, which is nice. But my first slide says exactly what you just said. And the only additional information was I've been there since my current post since 1998. In fact, Daniel and I were colleagues together for many years.
Do we have those slides up? I'm not sure if we do as yet, unless they're there in the background.
Seeing as I'm eating into my 5 minutes, I'll do my first slide from memory, which is that to say that CAR T cells are living drugs. That means they are T cells which not only get into the tumor, next slide, and the slide after, please, not only get into the tumor and start killing it, next slide, but are also capable of proliferating, expanding, and making copies of themselves, which leads to enhanced functionality. I'm looking for a point, but never mind. So there are various approaches to doing this, but the most common approach used clinically is what we call autologous. In other words, a patient's own. So blood is taken from a patient and T cells, which are the killer cells of the immune system, are extracted. And this here depicts a virus, which is essentially going into them and delivering a new gene. That new gene transfers the T cells into being reactive specifically with the tumor. These are grown up to large numbers and administered back into the patient. The idea being that through the bloodstream, they then get into the tumor sites. And in the tumor sites, they increase in expanding number, hence the concept of a living drug. Now, the specificity, which is conferred to your own T cells, is really determined typically by an antibody. This is the structure of an antibody here. And this end of it is the bit which recognizes things as parent. So an anti-GD2 antibody, this component is the part which recognizes GD2 on the surface of a target cell. So the trick is to combine those two bits of an antibody molecule into a single molecule on the surface of our T cell. So this is what the virus was encoding. And then the reactivity of a CAR T cell is actually determined by the bits inside, of which there are various combinations in which provide complicated signaling patterns. And essentially, it is the configuration of these combinations which confers that specificity. Now, the more signaling you put in, potentially the more toxicity will be. And so the state of the art is really balancing the specificity conferred by the molecule on the outside of the cell and the degree of activation which is conferred by what is inside it. So in other words, there's a lot of mix and match and plug and play. Next slide, please. And so in terms of where we are, in the leukemia and lymphoma field, in other words, the hematological cancer field, there are now 7 CAR T products which have received approvals by the FDA in the United States, but there are none for solid cancers. And the reason for this is that solid cancers are more technically challenging for CAR T cells. So solid cancer involves this very large accumulation of cells, not only tumor cells, but also immune cells. And many of these immune cells actually fight off T cells. One of the reasons solid cancers are successful. Now, in targeting of GD2, however, despite all these drawbacks caused by the inhibitory nature of the tumor environment, there is significant success. And I mention this as an introduction to Francesca, who's coming after me. And indeed, neuroblastoma is probably one of the forefront candidate diseases to benefit from this technology, not just of GD2, but because of other possible molecules that can be targeted on the cell surface. Next slide, please. And so, which CAR for which patient? Well, one of the most important concepts to get across is that each new product, each time we make something new with a new targeting against maybe a new cell surface molecule and a new configuration of all those signaling bits on the inside, we really need a separate phase 1 safety evaluation. And because within solid cancers in pediatrics, we really haven't yet identified the configuration which is going to give the sort of success seen in the leukemias. This means that there are many studies going on. And so there's really a pressing need for international collaboration. And these studies really have to be driven by academics because these are small numbers of patients and industry has not invested in CAR T development specifically for children's cancer. But what we really want is a toolkit of different CAR T designs for different patients so that we are able to expand the populations of patients who can benefit. And within the next few years, there'll be a range of augmented types of CAR T cells targeting these different antigens, which I list here. Next slide. So I hope this has been a suitable introduction to my esteemed colleagues who follow after me. And we thank you for your partnership and commitment to change the face of children with neuroblastoma. Back to you, Daniel.
Thanks so much. Impeccably on time, so congratulations for that. We're going to move on to our second speaker on CAR T-cell topic, who is Dr. Francesca Del Buffalo, who is from the Bambino Gesù Children's Hospital in Rome and has been very closely involved in the CAR T-cell project that they have there that was relatively recently published and has caused a lot of excitement. So Francesca, over to you.
Thanks a lot, Daniel, for this introduction. I will be very quick in this part. I'm a pediatric hematologist oncologist, and I work at the Bambino Gesù Children's Hospital in Rome, where I mainly manage our academic CAR T program for different oncologic and hematological indications. And in neuroblastoma, for sure, our focus has been dedicated in the last years in the development and testing of an autologous third-generation CAR T approach. John just explained you the different possibility of construct that you can have in the CAR T field, while we decided to include the two co-stimulatory molecules to enhance the power of the CAR T approach. And we are targeting as tumor antigen the GD2, which is expressed on a high proportion of patients with neuroblastoma on tumor cells. We have activated back in 2018 a phase 1/2 clinical trial, which was completed a couple of years ago. And the very first aim, as John was explaining, of this study was, first of all, to understand the therapeutic potential and the toxicity of this approach. And so we initially aim to understand what was the safety profile of the treatment and what was the highest dose that we could administer to the patients. And what we observed with this very first evaluation of our construct was that it is characterized by very specific toxicities, including hematological toxicity, which is for sure the most common and the most severe toxicity. But we also have mild CRS in 80% of the patients, and up to 10% of the patients, we also observe neurological toxicity. So it's a product that has its own peculiarities and needs some expertise to be managed. All the toxicities were transient, and none of the patients experienced permanent sequelae. And we defined the recommended dose of the treatment at 10 million of transduced CAR T cells per kg of recipient body weight. Despite the trial being closed, considering the high demands that we were experiencing and the timing needed to start a new clinical trial, we continue to treat the patients under a hospital exemption setting in Rome using exactly the same criteria that we were using in the clinical trial. But at the same time, we are working very intensely to start the new clinical trial, which will be a European multi-center clinical trial. Next slide, please. In terms of efficacy outcome, we have now been able to analyze an initial cohort of patients that can give us some more information and insights on the efficacy of the approach beside the toxicity that I just showed you. And what I can tell you is that 77% of the patients that were treated with active disease showed a response to the treatment, which could be either a partial response or a complete response, indicating an activity of the CAR T cells against the tumor. And not only this, the response can be durable in a proportion of patients, in particular those patients that are treated with 10 million of CAR T cells per kg and with a low tumor burden, so with a disease that is not highly spread. The probability of maintaining a survival without disease at 5 years is now in the order of 53%, so half of the cohort. But not only this, we evaluated also what aspects could impact positively on the response of the treatment, and especially on maintaining the response, which is to us one of the most important aspects of an effective and successful treatment. And what we observe is that if we treat the patients earlier, after the failure of 1 or 2 lines of therapy, the probability that the CAR T can induce a durable remission of the disease is significantly higher than patients treated with a more advanced disease after failing more lines of therapies. Not only this, we also consolidated the proportion of patients that had extremely high risk of relapse at the end of the first line, despite being in non-evidence of disease. And we have seen promising results, although these are very early results, and we need to enlarge the cohort of patients and prolong the follow-up of our patients. And one last point that we observed with our autologous approach is that the healthiness, the fitness of the lymphocytes that we use to generate our CAR-T have an important impact on the efficacy of the approach. And indeed, when we generate CAR-T cells from lymphocytes collected at diagnosis, this is associated with higher long-term survival. And this suggests, again, the importance of the fitness of these T cells to generate our drug products. Next slide, please. And as a challenge, I was asked also to introduce the concept of donor-derived CAR-T cells, because this is an approach that in the future may give us even more promising results and may be more accessible and more exploited in neuroblastoma patients. And if you consider the last point that I just touched, meaning the importance of the fitness of the lymphocytes from which we generate our CAR product, the idea is to use a donor derived lymphocytes, meaning lymphocytes derived from a healthy person. Usually, it's one of the parents of the child that has neuroblastoma, to generate our CAR-T cells. These are allogeneic donor-derived CAR-T cells. We have tested this type of approach in a very small preliminary cohort of patients, and what we have seen is that in some of them, allogeneic CAR-T could induce responses even if all the other previous treatment, including GD2 CAR-T autologous, had previously failed. This tells us that this approach may be relevant for a cohort of patients, and we also further characterize the drug products, the CAR-T, to understand what features could these cells have that improve the activity of their activity. And we observed some improved features that tells us that in the future this approach is really warranted further evaluations. But of course, we need more studies and further experience to understand the true potential of this approach. Next slide. And with this, I thank and the organizers for this incredible workshop. I thank you all for your attention, and I'll be happy later to engage in the discussion with all the other esteemed colleagues.
Thank you so much. So to complete our 3 talks about CAR-T, I'm going to reintroduce Robbie Majzner, who is the director of the Pediatric and Young Adult Cancer Cell Therapy Program at Farber and Boston Children's Hospital. And Robbie, you also have 5 minutes. Thanks very much.
Perfect. Hi, everyone. Thanks, Daniel, for the intro, and thanks to everyone for being here. So I was asked to speak about just the challenges of CAR T cells in solid tumors, and then maybe what we'll see emerge over the next 10 years as these hopefully become part of the standard care for patients. And just as an introduction, I am a physician scientist and run a lab that's focused both on CAR T cell engineering, but also on the ganglioside target GD2, which everyone knows is well from its centrality to the treatment of neuroblastoma. Go to the next slide. So why are solid tumors harder to treat with CAR T cells? So as people may know, CAR T cells really first emerged in hematologic malignancies, and in pediatrics, that's basically been in B-cell acute lymphoblastic leukemia, where CD19 CAR actually can render 80 to 90% of patients into an initial remission, which is really just unheard of for patients with any disease at all, but especially with a multiply relapsed disease. And this has quickly become part of standard therapy for patients with relapsed leukemia. But there's challenges as we think about going after solid tumors. First of all, when you go after a hematologic malignancy like B-cell leukemia, you can go after what's a lineage-derived target, or a target that defines that type of cell. And since the cell comes from B cells, and CD19 is expressed on B cells, which is a subtype of immune cells that fortunately you can live without, we can give a CAR that recognizes this highly expressed target that's on leukemia, that is only on a type of blood cell that you can live without. So patients lose their B cells, but they can live without it. That question is much harder for solid tumors. If you think about a solid tumor that starts in a nerve, you can't go after something necessarily that's on every nerve cell. And so there's been this search for targets, and many of the targets that we've ended up with, maybe GD2 is an exception, are targets that are expressed are clean on normal tissue. So they're not on the heart, they're not on the lungs, they're not on the liver, but they're expressed at pretty modest levels on the tumor. And not only are they modest, it's different from patient to patient and even from tumor cell to tumor cell. And sometimes you get a biopsy from two different parts of the body and the level can be different. So finding the right target or combination of targets is hard. T cells, it's hard for them to get into a solid tumor. And the best evidence for this is that we see patients have a response to GD2 CAR, but also other CAR T cells and other diseases, generally when they have disease that's in the bone or bone marrow, which is much more accessible than something that's within a solid mass. And then finally, solid tumors are just really good at suppressing natural T cell immunity. So they can secrete things called cytokines, which are proteins that inhibit the function of immune cells. And they even have other immune cells that can come and kill anti-tumor T cells or other anti-tumor cells. And it's hard because we don't always know which of these is at play in any specific tumor. You go to the next slide. So how can we overcome each of these things? So for the question of antigen heterogeneity, this is this issue that tumors can figure out how to live without almost anything. We expect, like we've seen in leukemia, that tumors will lose one target. So we know GD2 loss is a mechanism of escape to anti-GD2 antibody. We've seen that in patients. The data is still emerging whether or not that's happening with CAR T cells, but it definitely happens in mouse models. And so we really need to think about targeting more than one of these targets at a time. And that's hard because you need to find a target, both of which are on tumor cells and maybe on different types of tumor cells. So you knock out one, you still have another target that might be on a different type of tumor cell. But those targets can't be on the same essential normal tissue because then you would have even more toxicity against that normal tissue. The next thing is about trafficking. And so you basically can endow your T cells with anything that you think of because we make these in the lab. And so people have thought of all types of things, such as methods that the T cell really can cut through tough stroma that's around the solid tissue. But none of these have yet been tested in the clinic. And then finally, I mentioned this idea of suppression, that there's cytokines or other immune cells that suppress T cell activity. And here you basically can what's called armor your CAR T cells or enable them to avoid those suppressive cytokines or even sometimes take an inhibitory signal, a negative signal, and turn it into a positive. Some of these have already shown some fruit in the clinic, both in adult and pediatric cancers outside of neuroblastoma. And I think one challenge that scientists and researchers have right now is there's just so many ways to do this in a dish and in mice. It's hard to know which one to bring to trials because we don't always know why our therapies aren't working when they don't work in patients. Go to the next slide. And then finally, I was asked to share, what could things look like if CAR T cell would become part of the standard of care? Some of this is probably going to happen, and some of it may be more lofty, hopeful goals. I think one thing we really need to do a better job at is understanding antigen expression on our patients. Many patients who come for CAR T cells will have already received multiple rounds of anti-GD2 therapy. So if they're getting a GD2 CAR, it's important to know if they have that potential to benefit. And if the GD2 is not there and they can't benefit, like they're doing in Rome right now, they should be sent to another potential therapy. But really, eventually, you could imagine where we have several of these therapies and you give a mixture of therapies or a product that goes after a mixture of antigens that's really suited exactly for that particular patient. Like Francesca mentioned, collecting cells earlier from patients, either at diagnosis or after a small amount of chemotherapy, just to get the patient in a better state before collecting T cells, may be better because we may end up with healthier T cells. And hopefully, as this therapy advances and is proven to have benefit for patients, a real goal needs to be finding the patients who this benefits. That, of course, we want to cure more patients, but also we want to reduce the price of therapy, or the secondary the long-term effects of therapy, by trying to eliminate or reducing other types of therapy, including potentially anti-GD2 antibody and of course chemotherapy, autotransplant, etc. And then I just want to note one thing that's happening in the CAR T cell field, and there's been a few companies that have put out really exciting data lately. So as you guys know, and John mentioned, CAR T cells are made outside of the body. So a patient's T cells are collected and then they're transduced with a virus. A virus is used to transform them into CAR T cells. And there's now been patients that have had B cell cancers where instead of doing that, they've just injected the virus, and it's a modified form of the virus directly in the patient, so that, and it's a modified form that is targeted directly to the T cells. And to me, it seemed like a pie in the sky idea when I first heard about it, but actually the data has been quite exciting, with several patients achieving real responses. So these this virus is able to find the T cells, and the T cells are able to proliferate. So we may even be able to skip the step of making these outside the body, and this would make them much more widely available, but also way easier to run a trial. And way cheaper to run a trial, such that we could do even more trials and figure out more of how to best enable these therapies in patients. Go to the next slide. I don't know if I hit my 5-minute limit, but thank you for the invitation. I'm looking forward to the panel to come.
Thanks so much. Thanks so much, Robbie. Yeah, it's a really exciting perspective on the future. So everyone, hold in mind all of that information you've heard about CAR T. We're going to slightly shift gear, but we're going to come back to the CAR Ts in the discussion. So next, I'm going to introduce Shaquille Modak, who is the chief of the Neuroblastoma Service at Memorial Sloan Kettering Cancer Center in New York, who's going to talk to us about a new modality for imaging for neuroblastoma. Thanks, Shaquille.
Can you hear me?
Yep, we can. Thanks.
I'd like to first start off with thanking NANT for putting all this together. Amazing work. We looked after Sheer, and I want to remember him, and all these treatments hopefully can help other children like him. So I'm going to switch gears and talk about imaging of neuroblastoma. And next slide, please. As most people attending this talk know, that what's called functional imaging in neuroblastoma is critical. By functional, I mean that there are molecules on the neuroblastoma that take up MIBG and other molecules as well. And we can track these molecules in patients and then figure out where the neuroblastoma is. And thus far, over the last 25 years or so, MIBG therapy has become the gold standard. MIBG scans, are done by injecting a molecule called iodine-123 MIBG, which is MIBG attached to a particular form of iodine, and that emits gamma rays, and there it's imaged by a scanner. There are problems with 123 MIBG, such as some lesions we don't get very good pictures of, especially in the soft tissue. There's a lack of quantitation. We don't know whether a lot of MIBG uptake is worse than less MIBG uptake. It requires 2 days of imaging, and also can hurt the thyroid, needing thyroid blockers. Next slide, please. So we and other investigators replaced the iodine, as you can see circled here, with fluorine, and instead of MIBG, we made MFBG. And to summarize all the studies that have been done here, initially we did the first in human at MSK, also recently published on our phase 2 study, but other centers in Europe and China have also had access to this agent and have published on this agent. And recently, a multi-center pharma-sponsored phase 3 study was completed, the results of which haven't been published yet. But to summarize all of this, we know that MFBG provides much better images because we can do a PET scan rather than a gamma camera scan. we can quantify how much uptake is seen in each spot, and these spots are discrete and they can be counted individually. It's very patient-friendly. It's injected on one day, and 1 hour later, we have a PET scan, which can be done. And in the modern era of imaging, we can do very fast PET MRs. We don't need any iodine because iodine drops because there is no radioactive iodine in the mix. And most importantly, we are quite convinced that we find more lesions which are truly positive. Next slide, please. So in the phase 2 study, which we published recently, we had 37 patients. And in these 37 patients, all patients with positive MIBG scans also had positive MFBG scans. But in the MFBG scans, many more lesions were seen than on MIBG scans. In fact, 273 extra spots were seen. And we were able to follow 234 spots serially in 30 patients by doing regular scans as these patients went on. And indeed, in all these 234 lesions, relapse was detected by other modalities such as MIBG or MRI scan. So this one concern of MFBG detecting false positive lesions seems to be at least mitigated in this study. Next slide, please. And here you can see one patient where we did concurrent MFBG and MIBG scans, and we found on the left the MFBG being positive, which was negative on the MIBG scan. So we really didn't know what to make of this. But the patient then went on to a couple of weeks later to get an FDG PET scan and then a CT scan, and both of these showed bony lesions, suggesting that the MFBG picked up a true positive disease which was missed on the MIBG. Next slide, please. And in this, we have not published this yet. We show this one of my patients who had a positive cranial lesion in the skull on MFBG, as you can see in images A and C. When we did an MIBG on at the same time, image D, we don't see any uptake at all. 6 months later, unfortunately, a patient developed MIBG positivity, as you can see on the lower image, and then in image B, you can see that this was also positive on an MRI scan, again suggesting that we had picked up a true positive lesion. Next slide, please. So what do we do with all this information? First of all, we don't have, there is no access to this agent in an approved manner. There's still regulatory approval pending in the US. Although we strongly feel that we are picking up these true positives, we need to reproduce this in multi-center studies. And if we all switch to doing MFBG scan, it will have major impacts on how we stage patients, how we assess the response in patients. But most importantly, can we use this for earlier detection of relapse? And in 37 patients we've imaged, only in the one patient I showed you was completely negative on MIBG scan, but detected an MFBG uptake. We have not imaged many other patients with evaluable disease, so that really needs to be looked at. And all these areas need to be sorted out before we can use this agent in a widespread manner. And so I'll stop there and thank you. Next slide, please. Again, I thank Inat for really doing a yeoman's job and putting all this together.
Thank you. Thanks so much, Shakil. And I'm just going to move us now straight on to our final panelist, who's going to talk about liquid biopsy and ctDNA. And this is Dr. Gudrun Schleiermacher, who is a pediatric oncologist and a scientist extraordinaire at the Institut Curie in Paris, and I think has just come directly off a flight to join us. So thanks so much, Gudrun.
Thank you very much for the introduction, and apologies for any airport background noise. And I am the chair of the SIOPEN Translational Research Committee and Biology Committee, and as such, a huge pleasure to speak about one of my favorite topics, liquid biopsies, to you this evening. Next slide, please. When we consider oncology, we know it is hugely important to determine biomarkers that might determine or refine diagnosis, contribute to the determination of prognosis or risk groups, or predict response to specific treatments. And classically, this is performed via different biological analyses on a tumor biopsy. Liquid biopsy refers to the detection of tumor-specific biomarkers or molecules in body fluids, most frequently in the blood, but also other body fluids, just such as a cerebrospinal fluid or urine. And this has huge advantages because it can be minimally invasive and, importantly, also enable sequential analysis, which by definition is not possible on biopsies on solid tumors, especially neuroblastoma, given their sometimes complex localization. It is important to know that different biological analytes, different molecules might be analyzed. And when we speak about liquid biopsies, often we refer to circulating tumor DNA. These are little DNA fragments that are released from the tumor into the bloodstream following different processes, and thus form a fraction of cell-free DNA that is present in any individual. However, other molecules might also be analyzed, such as cell-free mRNA, microRNAs, extracellular vesicles, circulating tumor cells, as well as other analytes such as educated platelets. So next slide. The analysis and development of liquid biopsies has really seen a hugely rapid development, especially in oncology, solid tumors. And firstly, a number of diagnostic labs now propose techniques that enable analyses of liquid biopsies in a molecular setting. This might be of importance at the time of diagnosis. And for instance, in neuroblastoma patients, little infants who might be too sick to undergo biopsy, or in case of inability to obtain a biopsy, such analyses on liquid biopsies can enable to refine diagnosis and determine the biomarkers that are important for treatment stratification up front. We all know of the importance of determining the MYC amplification status, other DNA alterations, or looking for ALK mutational status. Importantly, at the time of relapse, molecular profiles might be of importance within the context of precision medicine programs and contribute to the identification of targetable alterations, looking for ALK or other targetable alterations, and thus contribute also to the study of mechanisms of tumor resistance. Next slide, please. A bigger challenge in the field of liquid biopsy analyses is integrating this in the concept of monitoring disease, determining the response to treatment, or even seeking to look for evidence of disease in the absence of disease on imaging in the context of studying minimal residual disease. To develop such techniques, different approaches are also being developed in different collaborative groups, which need to balance out the different sensitivities and the specificity of these things, such as digital droplet PCR, large NGS panel sequencing, whole genome or whole exome sequencing. Importantly, it is important to remember that we can use these analytes to follow patients during treatment. And for instance, in the graph on the bottom on the right, we measured the ALK mutated allele fraction, the VAF, in sequential blood samples of a patient And we could see that we could see a rise in the ALK VAF in the blood of a patient at a time when the patient was deemed to be clinically and upon imaging in stable disease. However, relapse unfortunately occurred, confirmed by imaging a few weeks later. So we do not really know how to integrate these analyses into our follow-up of the patients, but preliminary observations do suggest that it might be possible to identify tumor-specific alterations prior to imaging evidence of relapse in certain conditions using specific techniques. Currently, the European trial Mona Lisa, led by Liva Titgat and Sabina Taschner, seeks to determine in a randomized trial the feasibility and the clinical benefit of these approaches. My main sentence at the end of this talk is to insist on the importance of collaborations as these techniques are being developed. I think it is hugely important that we exchange expertise and data sets to determine how to integrate this knowledge into monitoring and follow-up of our patients. Thank you very much again for the invitation to contribute to this panel.
Thanks so much, Gudrun, and thanks to all of our speakers. So if we can get everyone back on camera from our panel, we're going to try and integrate some of that now into sort of one conversation of how we might put all of this together. So let's just start by thinking about this MFBG imaging. And Shakil, I think it's clear that the MFBG is picking up more lesions than standard MIBG imaging. And I guess the challenge is how we're going to know what to do with that in reality. And would you suggest that if we picked up disease only on MFBG, that would be enough to say that that patient had a relapse and we should start treatment?
The clinical experience we've had is so far only in 2 patients. That is, only in 2 patients were no lesions seen on MIBG, and a lesion was seen on MFBG. And in both these 2 patients, it was real. In the other patients, even though 273 extra lesions were seen, some lesion was seen on MIBG scan. So it was not a yes-no change. Having said that, I kind of have come to the conclusion that this is real, and one could focus on these lesions on MFBG. Does require further corroboration, but one could monitor these lesions more carefully and perhaps even think about radiating them, for example. But our experience comes from a small number of patients so far.
Okay, thank you. And Gudrun, to put a sort of similar question to you, if we had a patient who had no evidence of disease on imaging, perhaps including MFBG, but their ctDNA showed detectable tumor, do we know what to do about that at the moment? Do we know how to interpret that?
So a quick answer, no, we do not. A longer answer, it's a hugely important question and we need to work together to figure it out. It depends on a multitude of factors. First, on the technique being used, its sensitivity and specificity, on the dynamics of different cell-free DNA markers. To give some numbers, if we're looking at ALK and we had a variant allele fraction of 2 and it goes up to 2.2, that might not be significant. If we had not seen anything, but it goes up to 20, that might be. It cannot be disputed that for a given child, maybe in some instances, this would already enable us to conclude to start specific treatment depending on what the alteration is that we are detecting. But overall, I think, especially when we're considering these techniques also in view of early clinical trials, it is important to standardize the readout, the cutoffs, what we're looking for, and determine this also in the context of internationally validated neuroblastoma response criteria, so that we speak a common language also when we use this for treatment decisions, especially in the context of early clinical trials. So to go back again to the question, no, today I do not think that we can answer this question easily.
Thank you. And I think, to put this into context for our CAR T-cell colleagues, coming to them now, this issue of minimal disease and treating patients with CAR T-cell setting in that setting of minimal disease, not of bulk soft tissue tumors, not of widespread disease, seems to be becoming increasingly important. So I guess the question is, do we have any sense at the moment about where CAR T-cell therapy is going to fit best? Is it going to be in patients whose disease is pretty much disappeared on imaging, but maybe we've got a little bit of detection on ctDNA? Or is it going to work better for patients with more disease? How are we going to work out how much disease is the right amount to use for CAR T-cell patients? I don't know who wants to tackle that one first. Robbie, maybe you. You're in the middle of my screen.
Sure. Yeah, I mean, I think it's a good question. I think if you look right now with Francesca and the group in Rome's really beautiful data, it's working for patients that have small amount of disease. And this isn't completely surprising because all of the data from leukemia says that CAR T cells work better in patients that have small amounts of disease. And it's really, if you send a bunch of soldiers in and they have to defeat fewer enemies, they're going to have an easier time. So probably with the current generation of CAR T cells, whether that be for leukemia or for neuroblastoma, it's probably better to do it when patients have less disease. But T cells are capable of getting rid of huge amounts of disease. And we see this in examples from adult cancers like melanoma, where patients get checkpoint inhibitors or something called tumor infiltrating lymphocytes. These are able to get rid of really profound amounts of disease that is distributed throughout the body, lungs, skin, everywhere. And we've seen that even with sarcomas in a few cases. So T cells and CAR T cells should be capable of mediating those types of responses. I just don't think we're there yet with our current generation of engineering. And I think what we have right now still can have really great benefit for patients and potentially be a better therapy for end of chemotherapy, post radiation, post surgery, for getting rid of minimal residual disease that's maybe more effective than anti-GD2 antibody or pairs with anti-GD2 antibody. We don't know. I think we have to be open to the fact that, or admit the fact that we don't know, and then say we need to do clinical studies really to solve this problem.
Yeah. And Francesca, for the current studies or the current patients that you're treating in Rome, what sort of thresholds are you using to say how much is the right amount of disease?
Yeah, thanks for this question. So we have set up a threshold with the first set of patients that we treated, which showed us a difference in the outcome. And this is what we call the low disease burden threshold, which means 50% of infiltration in the bone marrow with neuroblast, skeletal involvement using the European SIOPEN skeletal score of 7 or below. And in case of presence of solid masses, the longest diameter needs to be below 5 centimeters. But what is important to note is that this is what we found out in our first cohort of patients. This is not written in the stones. Maybe in the future we will further refine this threshold with an enlarged cohort of patients. As Robbie was saying, technologies may improve, and so this may change in the future. At the moment, this is what is giving us the best results. And so our aim is to bring the kids to CAR T with this threshold. I wanted just to add one comment to what Robbie said, which I quote completely, which is also the toxicity point of view. So the less the disease, the less the activation of the CAR T, and so the less the toxicity that the patient experiences. So this is another pros to bring the kids to CAR T when the disease is under control as much as possible.
And John, to sort of follow up on that with you, because you're involved in developing multiple new CAR T cell constructs, do you think there's a danger that we are only likely to test those in patients with very aggressive advanced disease and a lot of disease? How do we select the patients for those early phase trials of new CAR Ts?
Can I make an additional point and then address that? I really like all the war metaphors, which often get used with CAR T cells. We've got tanks and armored cars and things, and Robbie makes a great point. But, you can't put a small army in to defeat a large opposing army unless you have a reasonable number of reserves. And so one of the concepts, of course, is that the numbers change. If you've got a rapidly growing tumor and you get some active T cells in, then by increasing those numbers, you can shift the balance again. And one of the concepts, an additional military concept, which is often used, this idea of armoring. By armoring, we're meaning, beefing up the T cells so that they produce a lot of the cytokines, and cytokines are a molecule which cause the cytokine release syndrome, which Francesca alludes to, which can, if uncontrolled, create the sort of very severe side effects, which can even become life-threatening. So within that context, it's clear that new augmentations such as this stand great potential for improving things, but also huge risk of disease. So it's always going to be evaluated initially in phase 1 studies within centers which have the experience really to know how to manage such potentially severe toxicities. And so I really do increasingly think that the small volume setting is the safer setting, not just because probably the patients are going to benefit more, but I think with this augmentation, there is probably enhanced risk of uncontrollable toxicity, and especially in a patient who has a high disease burden and potentially symptomatic from the disease burden. If you compound that with an infective CAR T cell, you can make an end-of-life situation worse. So it's a very difficult, of course, but that's a rather long way to answer your question. But I think some selection towards lower disease burden at the time of even going to phase 1 is the right way to go.
Yeah. So we may actually need to change the setting that we end up using these CAR T cells from the ones in which they've originally been developed to try and move them earlier in treatment, by the sounds of it.
Exactly. Yeah. Once it is established that something really works and has this capacity to shift the balance, as it were, as we describe, and it could be brought further forward and not be confined to the consolidation setting.
Yeah. And Robbie, you talked about one of the mechanisms of resistance being loss of antigen, loss of the target. And Gudrun, just to kind of put you on the spot, do you think there's anything from either ctDNA or similar technologies that could be used to help select patients or to monitor patients for that sort of escape mechanism as compared to how much disease burden they have?
I think indeed we are starting to explore the possibility of using cell-free DNA and the circulating tumor DNA as an indirect marker of disease burden, but we know that this is very variable for one patient to the next. So I think this is very early days yet. And the other context for liquid biopsies is that we might use more sophisticated techniques to look for the presence of the antigen itself that we are targeting to know whether it might be present or not. But here again, technological advances are still required to come up with homogeneous propositions to explore this, for instance, GD2.
Thanks. So unfortunately, we don't have a huge amount of time, but let's just kind of take a very kind of long-term perspective of 5-10 years time and see where we think we'll be. I mean, Shakil, what do you think is the future for MFBG PET? Do you see it as being the new replacement for standard PET or for MIBG imaging at some point down the line? Is that realistic? Oh, you're muted.
Sorry about that. If you ask any nuclear medicine physician, they're chafing at the bit to start this, get going, because it makes their life much easier in terms of, especially in terms of soft tissue. There's definitely a quantitative element to this. So I think if regulatory issues can be sorted out, this should be the best functional imaging modality we have by far.
And then we'll have to work out how to actually make it available to
everything else will
everyone
have
who
to
needs it.
be will have to change in terms of INRC and things like that, right? So, but nuclear medicine, everywhere can't wait to get their hands on this.
Yeah. And to our CAR T cell colleagues, if you could predict where CAR T cells would be in 10 years' time, how will you be using them in neuroblastoma? Robbie, do you have anything?
One of the really interesting questions, which has been debated within the SIOPEN group, is the replacement of high-dose therapy in the post-induction low-volume disease setting. So that's, in my view, something which should be evaluated scientifically.
So actually, it might be possible to use CAR T cells instead of high-dose chemotherapy to provide that consolidation.
Yeah.
is the thought. Yeah. Robbie, Francesca, do you agree that's the most likely best setting, or are there others?
I do.
Francesca, I'll let you go first.
On my side, I do, and I've been working with John and with the SIOPEN group a lot on this, so I cannot concur more. So surely this is one of the most promising, and I would say one of the things that I hope for the most, because we all know the issue of the high-dose chemotherapy. But then I think that there are other points where the CAR-T will go. I truly believe that we need to consolidate better those patients that have a very high risk of relapsing. We still need to probably define better who these patients are, but we know that there are. So that's another important point, and we are seeing these. And then we definitely need new technologies, armored technologies to overcome the issue of the disease burden. So I think these are the three main lines in which. And in the farther future, I truly believe that in vivo generation of CAR-T may change the field.
Wow, yeah, that would be
more than 5 years.
That would be a real game changer. Okay, but more than 5 years. Okay. And Robbie, from your perspective, do you see the same sort of direction?
I think it's dangerous to make predictions. This field.
Well, this is being recorded, so we'll be watching back on this in 5 to 10 years' time.
The field is just totally unpredictable. I mean, the first time I saw that in vivo CAR T cell suggestion, I just go, this is never going to work. But there's so many reasons it's not going to work. But it at least in early stages, there's the proof there that it can work. So I think it's dangerous to make predictions with too much certainty. I do think looking at CD19 CAR from leukemia, the low disease burden is a great place to think about using this. And another example from CD19 CAR. So for patients that have autoimmune disease, for a long time, people were using an antibody against B cells to eliminate those, and it helped reduce the symptoms of autoimmune disease. And then a group in Germany started using CAR T cells, which give you much deeper depletion than an antibody of B cells. And there have been amazing responses and maybe even cures of patients with lupus and other autoimmune diseases. So I also think that right now we use an antibody, and it's a very impactful anti-GD2 antibody, but this could, maybe it can't eliminate bulk disease like chemoimmunotherapy or like high-dose chemotherapy, but it definitely can travel through the body and sop up disease. Francesca has shown really nicely that it can go to the brain and find residual cells there. So I do think the pieces are there that this could emerge as a treatment for, post-therapy consolidation. But obviously our dream is to see it really be able to replace most of chemotherapy. And this is happening with immunotherapy and hematologic malignancies. The most aggressive type of adult B-cell leukemia right now is treated with a small molecule inhibitor along with an immunotherapy, blinatumomab. So I do think a lot will change over the next 10 years, let's say.
Thanks. And Gudrun, just to wrap up, again, to put you on the spot to predict the future, can you imagine a world in the future where we are routinely testing patients who've completed treatment for ctDNA as the means of detecting relapse?
Absolutely. And I know it's being recorded. Again, liquid biopsies at the time of diagnosis are already established in most labs for the molecular diagnosis. Now, to use this for surveillance, I can really imagine that maybe in the future, I can't give a timeline, maybe not 10 years, maybe 2 or 3, performing a blood sample every time we do an ultrasound or a CT scan, and nuclear imaging might be part of the routine. But we need to work together to determine the best technique and to determine how to integrate that in our overall response and surveillance criteria.
Great. Thank you very much. So we're at time there, but big thank you to all of our panel members for joining us from around the world. Thank you so much. Thanks to everyone for their questions. I'll try and put some additional answers in the written responses to those we've not had a chance to get to. Thank you all so much. And so we're now going to turn to the final part of the meeting, and I'm very pleased to introduce Nicole Scobie, who is a patient advocate extraordinaire, previously actually still recently retired air traffic controller, and has been involved on the Patient Advocate Committee of ITCC and is now the chair of Accelerate, which I won't explain because I imagine you will, Nicole. I'm going to hand over to you. Thank you so much.
Thanks, Daniel. So I will share my screen if that works. Is that working? I guess it is.
It is, yeah.
Perfect. Okay. Well, thanks to begin with, I wanted to thank INAT for inviting me to do this, and also say, just have a moment thinking of her son Shir today on this very important and symbolic day, because it is because of him that we're all here today, and so for him I am grateful. So the last part of this presentation was really, oh, I lost my screen there for a second. Talking about how I became a patient advocate and how any other parent or survivor would like, or patient would like to become a patient advocate. So my name is Nicole Scobie. I'm a patient advocate. And the things that Daniel mentioned here that I'll go through maybe briefly in the presentation about Accelerate. I'm on the board of Coalition Against Childhood Cancer. I'm a member of ITCC, one of the founding members of SIOPEN. And it looks like I'm the only founding member of SIOPEN, but it's plural, that's not just me. And I'm on the Childhood Cancer International R&I committee in Europe. But most importantly, I'm a parent, and that's my son Elliot on the bottom right-hand corner with his cape, Super Elliot, that he did wear to go to the hospital and insist that everyone called him Super Elliot. So how do we go from being a parent to being a patient advocate? The road is to go from my child and then to children I know and then to all children. And that's the path that we take. We are parents, we're survivors, caregivers, and there are also representatives from charities. We have a lot of different attributes, different knowledges of the disease, of different diseases, lived experiences, and of course we all have an urgency for things to change. We can have different roles. We can be research advocates, policy liaisons, funders, mobilizers. And the picture I put here is an example. It's I'm there on the right. I'm in blue on the far right. I'm at the EU Parliament, and on the far left is the mom of a girl who has neuroblastoma, relapsed neuroblastoma. In between the two of us, there are very high-ranking pediatric oncologists, researchers, and some politicians. But the important person in this picture is the woman on the left, who is talking about her daughter. And what she's telling the audience, which is all the MEPs and politicians and people who've gathered there to hear her, is about having to fundraise to bring her daughter across the border to get access to treatment. And this is really impactful. This is where we can have huge impact. And it was, let me tell you, it was really heard, much more than anybody else. So why do we matter? Why are we an essential piece of the puzzle? Well, because we bring the patient and family voice into the prioritization of research. We can define meaningful benefit because we speak from the real world perspective. We build trust and trial engagement from families. We, as I mentioned, can advocate quite effectively for policy change and for improvements. And of course, we raise awareness and we can mobilize resources, but we're also translators. And what do I mean by that? We translate in several ways. For researchers, we can give them the real world experience of the disease, of the treatments, and our priorities. For parents, patient advocates also can translate how research works, give information, what are the challenges, how we parents, patient advocates can help researchers. And then for the public, we bring a face to a really difficult subject, but we can make it personal and real. And then for the political world, we elevate the voice of the community. Our role is expanding. As we become more and more involved, we bridge groups, we can bring different stakeholders together, including pharmaceutical companies and researchers and regulators. We can address difficult topics. And of course, because we are outsiders, because none of us chose this world, we think with an outsider view. We advocate for the unmet needs so we can influence research questions, participate in the trial designs, and we can even evaluate and communicate on results of studies. We're generally seen as unconflicted. The child is at the heart of everything we do. And we're more and more getting involved in collaborations, as already mentioned in some of the presentations, partnering with different consortia like Accelerate, the COG, ITCC here in Europe, to harmonize strategies on a global level. So Accelerate, I did want to touch on that because I'm the chair of Accelerate just since 11 months now. Accelerate has existed for about almost 15 years, and it's one of the organizations that is, I think, really going to making a difference, and it's because we're working together. So what Accelerate does, very briefly, it brings together the stakeholders, and we are all equal partners at the table. So there's patient advocates like me, academic researchers and pediatric oncologists, industry representatives, and the regulators together at the table equally. And the goal is to identify challenges. So what are the issues? What are the problems in pediatric cancer drug development? And then work together in order to find solutions and make an action plan. And if anybody wants to know more about Accelerate, of course, you can Google it, or I think we'll probably be sharing this presentation after. So the last slide is just, again, about patient advocates. We are not just storytellers. We're not here just for our storytellers. We're strategic partners, and we're driving policy-informed innovation. And the future of drug development and of innovation in pediatric oncology, it really depends on our ability to bring science, regulations, and compassion together and to align those. So our strength is everyone pushing together towards the same goal with one voice. And so on that, I'll end, and I would like to call on everyone once again to take the pledge. And I think if I stop sharing, you will see it again. If you haven't already, there it is. You can sign up. Annette has done a great job of getting people really motivated by this, and I think I will pass it to you, Annette. Thank you.
Thanks so much, Nicole. I'm going to sign out at this point and just say I'm going to do my best to answer the questions that are still outstanding. And so I'll try and do that in the closing minutes of the meeting. But otherwise, thank you so much to everyone for joining. And I'm going to hand it to Annette, who has organized all of this, for the final words. Thanks so much.
So thank you all. Daniel, thank you for doing a fantastic job here, managing and making this happen. I want to thank Ro and Lucas and Julie and John and Robbie and Francesca and Gudrun and Shaquille and Nicole, because you did an amazing job here. And I want to thank all of you, parents, nurses, oncologists. No matter who you are, only together we can do it. Thank you for joining us this evening, this day, whatever time zone you are. I really appreciate it. And thank you. Thank you again.
Thanks, Nicole. Thanks all.